Hi, I'm Kurosh, co-founder and chief data scientist at LinkLab, and today I'm going to show you how to use Funcnodes, the main product of the LinkLab, to analyze your data. After a successful installation of Funcnodes and running the command funcnodes run server, a new browser page will pop up, and this is basically the first thing you will see. So at the top there are some areas: worker, node space, and setting. Setting is responsible for the changing of the appearance of the window. It's a matter of taste, of course. So then you can close it, and then on the worker you have the basically all the features related to the worker. So the worker is what you are building currently at the current window. So for instance, the worker that I'm working on now is called demo worker. So if I press demo worker, you see it's activated. I can stop the worker, restart the worker, and make a new one. So by clicking on the left panel, so it's like toggled on and off automatically, and by clicking on the manage libraries, we can see a list of already available modules. So basically these are the highly used Python modules that we converted them in a way that are compatible with our Funcnodes. For instance, we know the NumPy, so this is a very famous data science analysis Python library. But we have now here Funcnodes NumPy. It's a version that is converted to node-based version of the original NumPy. By adding this, I can see that it's appearing here, transformed in a way that we can use it here in our node-based presentation inside the Funcnode. What do I need for starting a data analysis here is, first of all, I need a way to upload my data. So that would be done via Funcnodes files. So by adding Funcnodes files, I can see now I have these options. I have the option of the uploading a file, uploading a folder, and so on. So for instance, if I click on the file upload, this is a node responsible for uploading single data. By pressing the upload file, there's a way to browse inside your disk and choose the data. So imagine that I want to use TIFF image. Now the data is uploaded, but it is uploaded as byte. Whenever you upload a file in the Funcnodes, it is converted to a byte file. So now that means we need a way to convert this back. We have the Funcnodes-images to edit image files. Here I need a functionality to transform this byte to an image. So here I can see that there is an option, it's called from bytes. Now connecting the proper output to the proper input, which is this to this, I can see now the image is created. If I click on the thumbnail on the node, I can see image in its original resolution and size, and if I close it, I still have some small thumbnail of that image already in the corresponding node. Now imagine that I want to get rid of this information on the bottom of the image, which is kind of typical in some scientific imaging techniques. So for this case, I need to crop this image. I can now connect again proper output to the proper input, and then here by decreasing the from but I am cropping the image. So I can also see it here on the thumbnail. If I press on the thumbnail image, I can see the better resolution of that image. Imagine I need to know the size distribution of these nanoparticles. So in that case, I need a way to segment these particles properly. So for that, I have Funcnodes-microscopy. In the microscopy, we can see there are some options. The file that was uploaded was a TIFF file. So here, if I basically connect the file upload to the from byte node and convert it from byte to image, I can see the image as here. But if I need more information, for instance, metadata of these TIFF files, I can basically call this node and again connect the proper output to the proper input. Now I have the image, the same image as here, as an array, because the output of this specific node is that image as an array, and also the metadata of that TIFF file. In order to have a better look on these metadata, I need another feature. So if I add the Funcnodes-basics and then go to the dictionaries and go to the dict get, now I have the option to connect this dictionary of the metadata to the proper input. And here I have a drop-down list, all of the metadata related to the uploaded image. For instance, I am specifically interested to see what is the pixel size. I can even search it. So it is like pixel size in nanometer. Here, if I click on that, I see the output, which is 6.075 nanometer. I use the TIFF file reader node from the Funcnodes microscopy. Back to segmentation of this image, there is an option in the Funcnodes microscopy in the image segment. So this node is responsible to segment objects within an image, but this node takes an image as an array. So I need to convert this to an array. So if I go back to Funcnodes-images, I can see there is an option to array. If I call this node and again proper output to proper input, now I can see that this returns array-like image. And if I connect this to the segment node and choose proper model, I can expect to see the segmented. So I use model from Stardust and connect the array image to proper input. This yellow is sign of specific node being active or running. You can see there are some outputs. What I can do is now to find a way to visualize, at least have a look by human eye to see if it's doing a proper segmentation or not. In this case, I need to use Funcnodes-openCV. This is another famous image processing Python library that we already converted into the Funcnodes-openCV, so it can fit in our Funcnodes ecosystem. So here, if I call for drawing, and more specifically, I wanted to draw contours. So here, draw contour node needs an image in its original image format, this part here or this one, or I can grab it even here and connect it. It also needs contours. So if I select this segment and if I click this part of the node, you see this node has, its name is segment node, of course. It has inputs. One is called image, which is this one. Another one is called model, and so on and so on. And there are outputs. The first output is contour. The second one is center. So I want to connect contours to the contours, and I can see there is something shown up in thumbnail. If I wanted to see it in better details, I click on that and I can see, yes, this image is well segmented, so to say. Now remember the first idea that we had. So we wanted to know this size distribution. In order to do so, we can have from the Funcnodes microscopy, we can select circles from contours here, and by selecting this, as we can see, it needs contours and centers. So contours were this one, so contours on this, and if I see now, it by default takes a pixel size within the image as 1 nanometer. If I want to be more precise, I can grab the pixel size from the metadata and connect it to this node. I can have precise size distribution in nanometer. So now if I connect centers to the centers, I can see, okay, there is now the output, and the output is in the dictionary. Now I have the option to use some functionalities of Funcnodes-pandas, which is basically a converted version of the pandas, to convert dictionary to a data frame. Go there, data frame, convert from dictionary, call for that, connect the dictionary. Now I have everything as data frame. I can see it as a table if I want to. So basically this is the information for each of the selected particles, which are now indexed as 0, 1, 2, etc. Around any segmented particle, a minimum circle and a maximum circle can be fitted. We have all the information for max circles and all the information for the minimum circle. So, and each of these minimum and maximum have their own properties. So the center of coordinates, the radius and the diameters, and also same for the minimum. But we also have the circularity score. We want to see how close they are to a perfect circle. So in order to visualize this information, the plotting features of the Funcnodes, that it's Funcnodes-plotly, and if I go down, I have the plotly here. I want to see the distribution of these information, so I will go for the plotly express 1D distribution histogram. So for histogram, I need a data as a data frame to be fed here, and then I can choose which of the columns of the data frame I want to basically see the distribution. Let's go for the circularity score. Now I can again see a thumbnail, and then if I want more information, I will click on that, and this is the distribution. I can also add the marginal to be as a box, so I will have a box plot on top of the histogram. So in this way, now I have more statistical information for circularity scores. So that means most of the particles selected within that image are around a 0.7 circularity score, 1 being a perfect. Now I can also have a look on the diameter distribution in nanometer. So for this, I can have distribution like this. So the median of the sizes of these nanoparticles within this image is like 86 or 87 nanometer, let's say. And these are being the outliers. Now let's upload another file. So now we are uploading a new file. As you can see, this image is a bit more tricky. So there are some highly dense nanoparticles within the image, even they seem to be on top of each other. Basically, we just uploaded the data. The whole pipeline of the analysis is the same. So even we can see here that the pixel size is already updated. It has a new pixel size, this image. And if we look at what the previous model did to this image in detail, we can see that it is doing segmentation, but it doesn't doing a good job. And even if we look at the size distribution within this segmentation, we see that we are sitting on the median of around 100 nanometer. But if we try another model, because here there are already different pre-trained deep learning models. For instance, if we choose suppose, we can see that this node is doing analysis because it is already active, and we are waiting for the rest of the pipeline to be updated. So yes, we now saw that it's updated, and now we want to look at the segmentation quality. So if we look now, we can see that this segmentation is much better. More individual nanoparticles are already selected, and if we want to see scientifically for our distribution, we can see now there is a huge difference statistically. So previously the median was around 100 nanometer, but now with a more precise segmentation, we get 73, 74 nanometer being the median of the distribution. So the same image, same nanoparticles of course, but different segmentation is providing different statistical analysis. So the quality of the pipeline and the models here are crucial. So that was another example of the image segmentation. And there are other things that can be done within this image analysis. You can also fit ellipses on particles. You can even fit difference on nanoparticles and get corresponding statistical information. So this is one of the functionalities or one of the features of the whole Funcnodes ecosystem. With that, I hope you enjoyed this video, and yeah, thanks for watching.